How do we record brain electrical signals? Have you ever wondered how scientists capture the electrical chatter of our brains? It's a fascinating process that reveals so much about how we think and feel. Let's break down the methods used to record these brain electrical signals. The most common way to record brain activity is through a technique called electroencephalography. In this method, small electrodes are placed on the scalp. These electrodes detect the brain's electrical activity, which is created when neurons communicate with each other. The placement of these electrodes follows a standardized system known as the International 1020 system. This ensures that they are positioned consistently across different individuals. Once the electrodes are in place, they pick up tiny voltage changes caused by neuronal activity. These signals are then amplified and converted into digital data for analysis. While electroencephalography is great for capturing rapid changes in brain activity, it does have limitations. The signals must travel through the skull and scalp, which can reduce the clarity of the data. For a clearer picture, there is another method called electrocorticography. This technique involves placing electrodes directly on the surface of the brain during surgical procedures. It provides much better spatial resolution, allowing for more precise recordings. However, this method is invasive and is typically used in clinical settings. If we want to look at brain activity at a cellular level, we can use microelectrodes. These tiny devices can be inserted into brain tissue to record the activity of individual neurons. There are sharp intracellular electrodes that penetrate the cell membrane to measure signals inside the neuron. There are also patch clamp electrodes that attach to the cell membrane to monitor activity without fully entering the cell. While these techniques offer detailed information, they are highly invasive and mainly used in research environments. Recent advancements in technology have led to the development of silicon-based probes. These probes can have multiple recording sites, allowing researchers to capture signals from hundreds of neurons at once. By integrating with complementary metal oxide semiconductor technology, these probes have become more efficient, enabling large-scale recordings in animal models during various behaviors. These recording methods play a vital role in understanding how our brains function. They help diagnose neurological disorders like epilepsy and contribute to the development of brain-machine interfaces. These interfaces can translate brain signals into commands for prosthetic devices or computers. In summary, recording brain electrical signals involves detecting the voltage changes produced by neurons. Techniques range from non-invasive electroencephalography to invasive microelectrode arrays. Each method has its own advantages and limitations, depending on the specific research or clinical needs.